This is my project. It's called Expression2Kinases Web, an updated web-based version of the Expression2Kinases pipeline. First discuss the goals a bit. So the original X2K, Expression2Kinases, was a 2012 project which produced an offline application whose ultimate goal was to help researchers identify upstream regulatory networks from differentially expressed genes. So the basic premise is you input a set of differentially expressed genes, and you run it through it, and then you get some idea about what is regulating those genes and causing them to be differentially expressed. The application combines 3 different tools, all made by the Mayan lab. So the first is ChEA, which focuses on transcription factors, so it identifies transcription factors that are related to your input gene list. Genes to networks, which looks through protein-protein interactions to try to combine and connect the various proteins that you input. And ChEA, which focuses on kinases, much the same way ChEA focuses on transcription factors. And X2K seeks to combine these 3 tools and together to allow you to figure out more about upstream regulatory networks. More on that a bit later. The goals of this particular project were to update the datasets used by the application, to then create a web app out of it so it's online and easily accessible so anyone who wants to use it can use it, and also to visualize the data directly in the app itself. The first step was updating the ChEA datasets, so the datasets that are used to identify related transcription factors. So the first thing was to just download the datasets from Enricher. Then afterwards, you can use a Python script, which I wrote, to rebuild the files into the correct format and also to clean out various entries which weren't needed. And use the following 4 different datasets, so the ChEA 2015, ENCODE 2015, ChEA and ENCODE consensus, and TransFac and JESCAR. All of them will be available as options on the web app. I'll show how that works in a second. The next step was to update the datasets for genes to networks, the second analysis tool. The raw data was obtained from ChIP-seq, which is this big hub of many different protein-protein interaction datasets. And the following 5 were selected to be updated: MINT, InATB, BioGrid, BioCarta, and Intact. And once again, a Python script was used to find all valid human and mouse gene entries, convert them, and remove the results from the high throughput studies that are sometimes found in these. Basically just to clean up the data and make it more usable and accurate. And once again, all of these different data sets, plus 3 from the old X2K web app, are available as options when you're customizing your analysis and then running it. And afterwards, this is just what it looks like. You end up on this web page. You'll see that you can input your genes in 2 different ways. You can either copy-paste them into this box or select a file containing them. This is the differentially expressed genes which you're trying to analyze. Afterwards, you have a whole bunch of settings where you can, on this particular tab, you can select which of the transcription factor databases, but you can similarly select which of the various protein-protein interaction networks, the sorting methods, and so on the other settings tabs. And then finally, you just run your analysis, selecting one of 4 analysis options. And that works as follows. The first thing is you have the 4 analysis options, but 3 of them are just the individual applications. So just the ChEA, which will allow you to see a graph of different transcription factors that are related to your genes. Genes to networks, which will find intermediate proteins and seek to build a protein-protein interaction network out of your input gene list. And ChEA, which will find related kinases and also display them in a graph ranked by significance. The final option is really the big one, the one which it's named after, X2K, where you run through all of them. The middle one should actually say genes to networks, but I'll get to that in a second. And it'll output a complete network. And here's how that works. Your input gene list first gets piped into ChEA, which will identify the set of top transcription factors, the ones most related to your input gene list, which will then be piped into genes to networks, where it will seek to find intermediate proteins that connect these transcription factors. Next, so once you have that network, that will get put into ChEA, which will seek to find related kinases. And finally, you'll end up with an output that looks something like this, where you can see that in blue you have your kinases, in green you're going to have your transcription factors, and in gray the other proteins. And ideally this should help give you an idea of what the upstream regulatory network is like in all of its different components. And the way the web app itself is actually structured is that we have a set of 4 different Java servlets on the back end receiving these various inputs, running the apps, so ChEA, genes to networks, ChEA, and the whole X2K pipeline. Then it gets sent back, and it's visualized directly in the browser using a site escape network for genes to networks, as well as the X2K pipeline, which looks nice, it's draggable, and chart.js for the various graphs for the ChEA and ChEA results. And you can also download the results yourself if you want to analyze them offline and take a look at what's going on there. I'd like to thank Dr. Avi Mayan, Greg Gunderson, who really helped out a lot with the project, all of the DCIC summer students, because it was a lot of fun, and they also helped, and all the other members of the Mayan lab, because everyone really helped out. It was great. Thank you very much.